morning, dear students. Today we are going to another section in translation. Yesterday I told you that today onwards we will start the different steps in translation that we are going to see. What are the required factors for translation? We know that various tRNA attached with amino acid is necessary. Then ribosomes, mRNA, it's certain that mRNA should be there, and different proteins that is performing various functions that also should be there. Various cations, then GTP for giving energy, all are some of the factors that is necessary for the translation process. Then which are the steps in translation? Initiation, then elongation, and finally termination. We will learn each step in a detailed way one by one. First is initiation. First there is mRNA, and in the mRNA we know that the nucleotide sequence are present, and among this nucleotide sequence, one sequence is important. Okay, and for translating this nucleotide sequence to amino acid sequence, we need the help of ribosome. So ribosome is coming and attaching to that. Actually, it is recognizing that codon, AUG. We have already learned that 3 bases form or 3 nucleotide form one codon, and that stays or that shows the beginning of a reading frame. From that point onwards itself, the ribosome need to start translation. Okay, that is the starting codon, AUG. Okay, then after starting from there, the ribosome goes on translating the remaining portion downstream. Okay. Then we are going to learn about the initiation of translation in prokaryotes. What are the processes? mRNA will be there, and first the ribosome is not coming and attaching the mRNA as such. As means as its organized form with the small subunit and large subunit. It's not like that. First, only the small subunit, that is the 30S subunit only is coming and pairing with that codon. It finds that or recognize that AUG codon. Okay, and for that initiation step in prokaryotes, some other proteins also necessary. They are called initiation factors 2 and initiation factors 3. And along with that, the RNA, one RNA in the 30S subunit is also very important. What is its significance, importance? I will explain to you now. Shine-Dalgarno sequence. This is a very important sequence that is present in mRNA. Okay, this is the mRNA and this is the starting codon. And from this starting codon, upstream between 5th nucleotide and the 10th nucleotide, that present a sequence that is called the Shine-Dalgarno sequence. What is the significance of that sequence? Yes, it is complementary to the sequence that is present, nucleotide sequence that is present in the 3' end of 16S rRNA, ribosomal RNA that is present in the small subunit. Because of that complementarity, that small subunit can recognize correctly and can proceed from the beginning of a reading frame. Okay, first ribosome recognize this Shine-Dalgarno sequence And after recognizing that, it can correctly locate over that start codon, and from that point onwards, it begins translation. Okay. Next is about the initiation factors that present in prokaryotes. As the name indicates, there are certain factors that help in the initiation of translation. In the case of prokaryotes, there are mainly three initiation factors that present. They are initiation factor 1, 2 and 3. This is ribosome small subunit, and this is initiation factor 2. It is bound with one molecule of GDP. Then came the initiation factor 3. What is the significance? Here you have seen that as it come and bind with the ribosome, or if this factor is present associated with the ribosome, it prevents the premature association of this large ribosomal subunit with the small ribosomal subunit. That is its function. Then will come the next initiation factor, that is initiation factor 1, that helps the small ribosomal subunit to go and bind with the mRNA. Yes, initiation factor 1, as it comes, it helps the ribosome to come and bind with the mRNA. Next is how the first amino acid tRNA is coming and attaching with the ribosome, that I am going to explain. And as this first tRNA comes and bind with the ribosome, it need to pair with the initiation codon. And initiation codon, we know it is nothing other than AUG. AUG is the initiation codon, and this codon, more than an initiation codon, it codes for an amino acid methionine. Okay, methionine. In prokaryotes, this methionine, it carries a formyl group. Formyl group means C double bond O H R, means this functional group. This is the formyl group. So in prokaryotes, that methionine amino acid, it bears a formyl group. So that amino acid, in the case of prokaryote, it is called N-formyl methionine. Why that? It is known as N-formyl, because whatever be the polypeptide chain, as a new polypeptide chain form, it starts from initiation codon itself. Initiation codon is AUG, and it goes for methionine, and that N is N-terminal end of that polypeptide chain. N-terminal of that polypeptide chain, and the amino acid get added at that end is methionine. And in the case of prokaryote, which type of methionine? A formyl. So that methionine is called N-formyl methionine. It get added to the N-terminal of a nascent polypeptide chain. After it get added to this polypeptide chain, later it is separated, removed enzymatically. Okay, then I will show you the image of N-formyl methionine. This is N-formyl methionine. This is the formyl group. This is the formyl group. C double bond O H R. That means that amino acid comes and bind here. This is the first amino acid that is adding to the N-terminal of a nascent polypeptide chain in the case of prokaryotes. Next, we are going to see different types of methionine tRNA. That means tRNA carrying that methionine amino acid. Two types of methionines are there. The methionine that is initiating the protein synthesis by get added to the beginning of polypeptide chain. That type methionine tRNA. They are they are different with respect to prokaryotes and eukaryotes. And coming to prokaryotes, that methionine tRNA that initiate protein synthesis is tRNA-F met, and in the case of eukaryotes, the methionine tRNA that initiate protein synthesis is tRNA-I met. Okay. Then there are some other methionine tRNA. What is its peculiarity? That methionine tRNA incorporate that methionine amino acid not in the beginning but the interior regions of the polypeptide chain, and those amino acid will not get removed from the polypeptide chain. It remain as a part of that polypeptide chain itself. That type of tRNA, methionine tRNA, also two types with respect to prokaryotes and eukaryotes. In prokaryotes, that type of tRNA is called tRNA met. In the case of eukaryotes, it's called tRNA met. Okay, this much all points are clear to you, I think. Next is initiation. How the translation is initiated in the case of prokaryotes, that we are going to see. Regarding to all these things, I already explained to you. Anyway, I'm just pointing out one by one. Initiation factor 1, initiation factor 2, 3, all familiar to you. I already explained to you. This is mRNA with the initiation codon. To that initiation codon position correctly, that ribosome comes and attached there. Then one new thing here I need to explain, that is 3 sites EPA, and each of these sites have some significance and different roles, and especially those roles are relating with giving attachment to incoming tRNA that carrying amino acid. Okay. Next, what happened? The tRNA that carrying the first amino acid, that is the methionine, comes and bind with the P site of the ribosome. Because we already understand that the methionine is the amino acid corresponding to the AUG codon. So that's why it come and bind at the P site. Okay. Then after it binds, then this initiation factors, initiation factor 1 and initiation factor 3 are released from the small ribosomal subunit. We know that initiation factor 3 is preventing the large subunit to come and bind with the small subunit. So as this initiation factor 3 is released, then large subunit is free to come and bind with the smallest ribosomal subunit. Okay. Then what happened? That GTP that is seen attached to the initiation factor 2 is hydrolyzed and release one phosphate group, and it becomes GDP. That means some amount of energy is generated, and as a result a conformational change happened there, and because of that conformational change, initiation factor 2 also released from the subunit there. Okay, that is all about the initiation of translation in prokaryotes. Next, initiation of translation in eukaryotes. In the case of prokaryotes, we have seen the role of 3 initiation factors, mainly initiation factor 1, 2 and 3. But in the case of eukaryotes, the number of initiation factors are more. Here 12 initiation factors are present. Then we need to understand what are the subunits of ribosome in the case of eukaryotes. In the case of eukaryotes, the subunits are 40S and 60S. Together it constitute 80S ribosome in the case of eukaryotes. As it came to prokaryotes, what are the subunits? 30S and 50S. Together it constitutes 70S ribosome in the case of prokaryotes. And just mentioning for your better understanding. Okay, that about the subunits. And here one more difference exists between prokaryotes and eukaryotes regarding the initiation of translation. What is the difference? In the case of prokaryotes, we have seen that first, that ribosome, smaller subunit, it came and with the help of some initiation factors, it first bind with the mRNA. mRNA is AUG, initiation codon, and later only that tRNA with the methionine is come and bind in the P site. That is in the case of prokaryotes. And I'm coming to the difference that is in eukaryotes. First, the ribosome smaller subunit binds with the tRNA. Here it binds with the mRNA, only later tRNA is coming. Here first tRNA carrying methionine is binding with the small subunit along with the presence of some initiation factors. And later, after that, after binding the tRNA, they together come and bind with the mRNA. That is the difference. This difference it will be more clear as I explain the process. Okay, that we are going to see the initiational process of translation in eukaryotes. Now, this is the ribosome, small subunit. Here I am showing only the P site now. And to this P site, the tRNA that carrying the methionine come and bind along with that eukaryote, E4 eukaryote, eukaryote initiation factor 2 along with the bound GTP. Okay. Then along with that some other initiation factors also come and bind. They are initiation factor 1 and 3. Okay. Then what happened? Then there is an important term, that is 43S pre-initiation complex. The whole structure, the whole assemblance, it is called as 43S pre-initiation complex. After forming this complex, it go and bind with the 5' cap of mRNA. After forming that complex, the whole assemblance go and bind with the 5' cap of mRNA. Okay. Then I am going to tell you about some initiation factors that already clustered in the mRNA. And what is the role of those cluster of initiation factors that present in mRNA? Those initiation factors help the 43S complex to come and bind to mRNA correctly. That is the role of the initiation factors that seen associated with the mRNA. Okay, one among such initiation factor, important one is initiation factor 4E. What it is doing? First, it come and bind the 5' cap and it carrying out a proofreading. It carrying out a proofreading in 5' to 3' direction, and wherever it found some secondary structure, means a folded or double stranded structure, it remove those portion for making the work of 43S pre-initiation complex easy. If there is such a structure, it is not easy for that complex to move forward. So, this initiation factor remove those structures and continues proofreading. And after that proofreading, that initiation factor hold the mRNA in the form of a circlet instead of a linear structure. What is the importance of converting the linear structure to a circular is not that much clear. Even then, I think that by holding it, it can prevent the action of other enzymes from degrading this mRNA. Maybe that a reason. Okay, anyway, it hold that mRNA in a circular fashion. After that, that 43S complex come and assemble here. Then its name changed to 48S complex. Then it moves along the mRNA and reaches AUG, initiation complex. And that GTP, that bound with the initiation factor 2, hydrolyzed to GDP. And after that, I'll generate some energy, conformational change, everything happen. And as a result, all these initiation factors are released from the ribosome or that 48S complex. As they release, then the large subunit come and bind the small subunit and everything well set. That initiation is happen. Okay. Okay, with this we can finish today's class and section, and next class we will go and see the other steps in translation. Okay, thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.